Hello, my name is Scott Mitchell, and I'm a PhD student in Sandy DeBano and David Wooster's Invertebrate Ecology Lab at Oregon State University's Hermiston Ag Research and Extension Center. While the ongoing COVID-19 pandemic has put a bit of a damper on our research, we're still getting out into the field and starting new research projects. The project that I'd like to talk to you about today and show you some of the things we're doing is a new project that Sandy and I are working on in collaboration with U.S. Forest Service wildlife biologist Mary Rowland. The project is taking place at the United States Forest Service Starkey Experimental Forest and Range located near Grande, Oregon. While we've worked at Starkey in the past, we've mostly worked in Meadow Creek, which is a riparian meadow system. However, this time our work is focused instead on a forested upland system around Syrup Creek. In this new project, we're interested in how the many different species of native bees that live in the forested systems will respond to an understory thinning and fuels reduction project that the Forest Service is starting this fall. Not only may projects like these decrease wildfire risk, but they may also provide benefits to wildlife and other animals like pollinators. Since the thinning is planned to take place in the late summer or fall of this year, we wanted to get out this spring and establish some study sites and sample the bee and plant communities before the thinning happens. To establish our study sites, we spend hours driving and walking through dense forest patches in Starkey, carefully evaluating the characteristics of each site before selecting those to use for further study. Once we had our sites selected, the fun really began and we could start sampling. Now, as I mentioned earlier, we were focused on sampling bee and plant communities, and to do that we use a couple of different methods. To sample bee communities, we're using 3 different sampling methods. The first method that we're using is called pan trapping. So to set up pan traps, we place 3 traps of assorted colors at 5 evenly spaced points along a 60 meter or 196 foot long transect. These traps are then filled with slightly soapy water and left out to collect bees. The fluorescent color of the traps that you can see here reflect light that bees see, and the bees are attracted to the traps as if they were flowers. And after we've left the traps out for around 48 hours, we come back and collect the bees that were caught in the traps. The second way that we sample bee communities is with vane traps, which are made of fluorescent plastic and collect bees in a very similar way to pan traps. That is, the fluorescent bright colors attract bees as if they were flowers. We place these traps on posts approximately 1.5 meters above the ground, and, as you guessed it, fill them with slightly soapy water. After the traps have been out for approximately 48 hours, we return to the trap and collect the bees that are in the trap. Since there's over 200 species of bees present in Starkey, and field identification is extremely difficult, if not impossible, we take all of our bees back to the lab, pin the specimens, and send them to a specialist who identifies the samples to species. The third way that we're sampling bees is a fairly new way for us. We use these nest boxes that we put out, and cavity nesting bees come and make nests in them. Later, when the bees hatch, next spring, we can identify those bees to species. Finally, we're sampling plant communities by laying out 5 20-meter transects at each site and counting every stem that we encounter that has a bloom on it. When we're sampling plants, Sandy usually counts the flowers while I record the numbers as she counts. This process can take a good while, but in the end it gives us a reasonable estimate of the resources that are available to bees at a given site. Of course, some sites have lots of flowers, while others have very few, so this can be quite a variable process. Next year, after the stands have been thinned, we'll return to the same sites and see how the bee and plant communities have changed. This project fits into one of our larger goals of our lab, which is understanding the effects of forest management on pollinators, many of which are active in adjacent agricultural areas. Stay tuned for future videos about bees and plants of Starkey and about this project. As we get some results, we'll keep on making videos to communicate.